Welcome back to the deep dive. Today, we're plunging into something, honestly, I think many of us really underestimate. It's this hidden energy factory right inside our own bodies.
Mhm.
We all sort of know the gut microbiome is important, right? But have you ever stopped to think about how much, actual energy it provides you?
Yeah, the actual quantity.
Exactly, or how that supply of fuel shifts, maybe dramatically, based on something as basic as what you eat each day. It's a really profound connection.
It really is.
Hmm.
And what's so compelling here, I think, is that we're digging into this really quite groundbreaking study. It's by Arnoldini and colleagues, published in Cell, I believe, in 2025. And their mission wasn't just, describing things. They wanted to quantify it, to actually put numbers on the varying harvest of fermentation products from the human gut microbiota.
Right, the harvest. I like that term.
It paints a picture, doesn't it? So for our deep dive today, our mission is really to understand how our gut microbes create these, well, vital energy molecules.
Yeah.
How much of them we actually absorb, and crucially, what factors, especially our diet, are the real levers, the things that control this metabolic exchange between, well, you and your microbial co-pilots.
Okay, so let's jump right in then. We're talking about these fermentation products. It sounds pretty scientific, maybe a bit like chemistry class.
It can sound that way.
What exactly are they, and why should we, listening right now, really care about them?
Okay, great question. So at their core, these fermentation products are mainly a group of molecules called short-chain fatty acids. You might hear them called SCFAs.
SCFAs. Okay.
Yep. And let's say the big three you'll hear about most often are acetate, propionate, and butyrate.
Acetate, propionate, butyrate. Got it.
Right. But there are others too, playing important supporting roles, like lactate, formate, and succinate. But think of them less as just chemical names and more like your body's hidden fuel source, and also its internal communication molecules.
Fuel and communication. Interesting. So where do they come from? How are they made?
Well, their origin is actually quite ingenious,
Yeah.
really. You can think of them as valuable byproducts, almost like gifts, if you will.
Gifts from our microbes.
Exactly. Gifts excreted by the microbes living in your large intestine that grow anaerobically. Anaerobic just means they thrive without oxygen, which is
Yeah.
perfect for the large intestine environment.
Right. No oxygen down there.
Precisely. So these microscopic factory workers, your gut microbes, they essentially feast on complex carbohydrates. Stuff from plant-based foods, think dietary fiber, resistant starches, things your own digestive system can't break down earlier on.
So the stuff that makes it all the way down.
That's the key. It has to reach the large intestine. Now, they do nibble a bit on undigested protein, maybe some of your own gut lining that's called mucin. But the vast, vast majority of their energy, their food, comes from those plant fibers and complex carbs.
All right, so let's maybe try an analogy here to make sure we're all following.
What do you?
You mentioned a factory. So my large intestine is a bustling, maybe microscopic fermentation plant.
Perfect analogy.
Yeah.
Yeah, fermentation plant. The raw materials coming in are those complex carbs, the fibers, the starches from your diet that survive the journey down.
Okay, raw materials in.
Then the diligent factory workers.
Yes.
Those are your gut microbes. Each type may be specializing in a certain task.
Great, the bacteria.
And their main output, the finished product, that's these fermentation products, the SCFAs mainly.
Yeah.
Think of it like a special, really high-value currency they generate.
A currency. I like that.
Yeah.
And then we, the host, we use this currency.
Exactly. You, the human host, absorb this currency and use it for a whole range of absolutely vital functions. It's not just waste, it's valuable.
Okay, that paints a much clearer picture. So once we've absorbed this microbial currency, what does it actually do? Is it just burned for energy like calories from food, or is there more going on?
Oh, it's much more than just fuel.
Yeah.
Yes, they are a direct energy source. That's definitely true.
Yeah.
Especially for these cells lining your the colonocytes. They absolutely love butyrate, for instance. It's their preferred fuel.
So direct energy for the gut lining itself.
Precisely. But beyond just energy, they have really significant regulatory functions all through your body. They can influence your immune system, helping to sort of manage immune cell activity, even call them to where they're needed.
Wow, okay. Immune function too.
Yep. And they play a role in satiety signaling. That means helping to tell your brain when you feel full. Obviously, super important for regulating appetite, right?
Definitely.
And they even impact the gut-brain axis. That's that critical two-way communication highway between your gut and your brain. So they can affect things like mood, maybe even cognitive function. It's complex, but the link is there.
Gut-brain axis. That's
Yeah.
a whole other deep dive, maybe.
It could be. And one more thing, they're also environmental shapers right there in the gut. By changing the pH level, the acidity, in the gut lumen, they help create the conditions that favor certain microbes over others. It's like they're tending their own garden.
So they influence their own environment. It's a whole ecosystem.
It really is a dynamic ecosystem. But the real breakthrough of this study, the Arnoldini paper, is that it moves beyond just, identifying these molecules or measuring their concentration at one point in time, like in a fecal sample.
Which is what a lot of studies do, right? Just measure concentration.
Exactly. That only gives you a snapshot. This study quantifies the flux, the continuous flow. How much is being made, how much is being absorbed, constantly. It's like moving from seeing, a single car on a road to understanding the entire traffic flow of a city throughout the day. That's the big leap here.
That makes sense. Understanding the flow, the dynamics. You mentioned this systems level approach earlier. Can you unpack that a bit more for us listening? What does that actually mean when you're trying to measure something so complex, so hidden away inside us? How do they even start to quantify this constant production and absorption?
Right. Systems level. It means they didn't just look at one microbe in isolation or one specific SCFA. They built a comprehensive framework. They tried to understand how all the different parts, the food going in, the different types of bacteria present their various outputs, the absorption, how it all interacts dynamically to create the overall energy harvest we get.
Seeing the whole factory, not just one assembly line.
Precisely. Seeing the whole integrated factory in operation. And to do this, their experimental foundation was incredibly solid, really robust. They started by meticulously growing many common gut bacterial species in the lab. I think it's 22 species, representing a big chunk of the typical gut biomass.
So recreating a mini gut in the lab almost.
In a very simplified, controlled way, yes. This allowed them to track very precisely what each species consumed, their uptake rates, and what fermentation products they excreted or released.
Okay, so tracking inputs and outputs for each bug.
Exactly. And a key insight came even from this stage, the sheer metabolic diversity. While many microbes were happy eating similar things, like glucose, their outputs varied wildly. Some, like bacteroid species, were primarily making succinate. Others, like those in the Lactospiraceae family, were churning out butyrate.
So who is in your gut does influence which products you get more of.
That's right. The specific mix matters for the type of SCFA. And interestingly, they found these production patterns were surprisingly consistent even when they changed the growth media or the pH levels within ranges relevant to the gut. This suggests these are pretty fundamental metabolic pathways for these bacteria.
Okay, so they studied the individual microbes in detail. That makes sense as a foundation. But the big challenge surely is scaling that up. How do you go from that lab data to understanding the total output in a real human gut, which is, vastly more complex?
Absolutely. That's the crucial step. And that's where the real ingenuity, I think, comes in. They use two completely independent ways, two different quantification methods to estimate the total daily fermentation product harvest in actual humans.
Two different angles to get the same number.
That's the goal. It's a form of scientific validation. If two totally different ways of calculating something arrive at similar answers, you can be much more confident you're onto something real.
Okay, so what were the two methods?
So method one looked at what was coming out, essentially. They used some really high-quality data from the 1970s in Great Britain, apparently unparalleled detail on daily fecal output. They estimated the bacterial biomass in that daily fecal dry weight, which is around 16 grams typically.
Okay, bacteria in poop.
Yep. And then they multiplied that biomass by their lab data, how much fermentation product a gram of bacteria produces on average. That gave them an estimate based on output.
Makes sense. Output-based estimate. What was method two?
Method two looked at what was going in. They estimated the total amount of microbiota-available carbohydrates, often called MCs.
MCs, like the fibrin-resistant starch we talked about.
Stuff that reaches the large intestine.
Yeah.
They estimated how much MCs would reach the colon based on a typical diet around 36 grams per day for that 1970s British reference diet.
Okay, based on dietary input.
Right. Then, using their lab data on how efficiently bacteria convert those MSCs into more bacteria, biomass, and then into fermentation products, they predicted the resulting product release. So an input-based estimate.
Two totally different approaches. One looking at waste, one looking at food.
Precisely. And here's the really powerful aha moment.
Did they match it?
Remarkably well. Both methods, despite being completely distinct, yielded very similar daily estimates for total fermentation product release for that referenced British diet, around 450 to 470 millimoles per day.
Wow. That's pretty convincing.
It really is. That agreement strongly validates their whole framework. It gives us high confidence in these kinds of numbers. And just to be super clear on one point, while things like dietary protein that escapes digestion and some host-derived stuff like mucin do contribute a bit.
The stuff the microbes nibble on.
Yeah, the study suggests they contribute maybe 20% at the absolute maximum to the total fermentation product release, and likely much less in reality. The vast, vast majority, we're talking probably over 80%, maybe more, of the nutrient supply for these microbes comes from those carbohydrates in your diet, the MACs.
So diet-derived carbs are king.
Absolutely. And here's another crucial finding about efficiency. They calculated that more than 90% of the carbon atoms from those MACs ends up incorporated into fermentation products.
90%. That's really efficient conversion.
It is. And even more striking, less than 2% of those fermentation products actually exit your body via feces.
Whoa, we absorb almost all of it.
Overwhelmingly, yes. What your microbes produce from your dietary leftovers, you absorb and utilize. It's an incredibly efficient internal recycling and energy capture system.
Okay, let's maybe take a quick pause here. We've covered a lot of ground. Just to recap the key points so far for everyone listening.
Good idea.
First, our gut microbes ferment stuff we can't digest, mainly complex carbs like fiber.
All right.
Second, this fermentation produces valuable molecules, mostly short-chain fatty acids, or SCFAs, like acetate, propionate, and butyrate.
Yep, the big three.
Third, these SCFAs aren't just waste, they're crucial fuel, especially for our colon cells, and they also act as signaling molecules influencing immunity, appetite, maybe even our brain.
Vital roles, absolutely.
And fourth, this study used clever methods combining lab work with two different estimation approaches, input-based and output-based, to reliably quantify the total daily production, the flux of these SCFAs, and found we absorbed almost all of them.
That's a great summary. Captures the essence of the first part.
So question for you listening, does thinking about your gut microbes as little factories churning out valuable currency change how you view digestion or fiber? Just something to ponder. Now this brings us to a really huge practical question. We understand how it's measured and what these products are, but what actually influences how much of this beneficial currency we get? Is it all about having the right specific mix of bacteria in our gut? Or is it mainly about what we're feeding them? This feels really key for everyday life.
That is the critical question, isn't it? And the study provides a very clear and perhaps for some, a surprising answer. Let's start with the microbiota composition, the specific mix of bugs. Yes, it absolutely varies widely between people. Everyone really does have a unique microbial fingerprint.
We hear that a lot, your unique microbiome.
And this study confirmed that these differences in species or families do impact the types of fermentation products you get. For instance, if you have lots of bacteroids, maybe you produce more propionate or succinate. If you have more lactanospiracine, maybe more butyrate. So the flavor profile changes.
Okay, so the mix affects the type of SCFA. Makes sense.
But here's the absolute key finding, the big reveal from their analysis. Variations in microbiota composition seem to have very little impact on the total daily release of fermentation products.
Wait, really? Little impact on the total amount?
Relatively little, yes,
Mm-hmm.
according to their models and data integration. So in other words, your specific bacterial recipe dictates the flavor of the fermentation output, more of this SCFA, less of that one. But it doesn't significantly change the overall volume of production coming out of the factory.
Huh. So the brand of worker doesn't change the factory's total output much.
That's a good way to put it. Which immediately leads us to the real driver. If it's not primarily the type of microbes, what is it?
It has to be the diet, right? The raw materials.
Yeah. Exactly. Diet plays the overwhelmingly dominant role. The amount of fuel you provide dictates the factory's output.
Wow. So if my specific gut bug mix doesn't dramatically change the total energy I get from them, then maybe all the focus we sometimes see on, say, taking specific probiotic strains to boost energy might be, well, maybe missing the bigger picture. That feels like a pretty bold claim against common narratives.
It is a strong conclusion from this particular quantitative framework, yes.
Yeah.
And the data they analyzed across different populations really illustrates this point powerfully. They looked at modern Western diets first, using data from the U.S. National Health and Nutrition Examination Survey, NADNEs, from 2017-2018.
Okay, so typical modern American diet.
Right, and they found a significantly lower median harvest of these fermentation products, around 286 millimoles per day.
Lower than what?
Lower than that 1970s British reference diet we mentioned, which was around 450, 470 millimoles.
So quite a drop.
A substantial drop. And this decline strongly suggests that our modern, often highly processed, lower fiber Western diets are directly reducing the amount of these beneficial fermentation products we get. Less raw material, less output.
That makes intuitive sense. Less fiber in, less for them to work on.
Precisely. Now compare that to the Hadza hunter-gatherers in Tanzania.
The traditional non-Western diet.
Exactly. Their diet is incredibly rich in fiber, lots of tubers, baobab fruit, other plant materials depending on the season. And their estimated harvests, much, much higher, up to approximately 1,000 millimoles per day.
Wow, like 3 to 4 times higher than the U.S. median.
In that ballpark, yes. And interestingly, their harvest showed strong seasonal variations, which directly track the changes in the types and amounts of fibrous foods available to them throughout the year. Diet changes, harvest changes.
A direct link in real time
It
almost.
really shows that connection. And they didn't stop there. They looked at broader global dietary data, again, mostly from around the 1970s, before Western diets became quite so globally widespread.
What did that show?
Even higher peaks in some populations, up to potentially 1,500 millimoles per day in individuals consuming certain traditional, very high fiber diets. Again, the common thread was the diet diverse, fiber rich.
So the conclusion seems pretty unavoidable then.
It really does. The big takeaway here is crystal clear from this analysis. Diet, specifically the amount of microbiota available carbohydrates, those MACs, the fiber and resistant starch that you consume, is the dominant factor determining the total fermentation product harvest. It far, far outweighs the specific mix of bacteria you happen to have living in your gut at any given time.
Your food choices are basically turning the main dial on your internal fermentation factory's output.
That's the message. Much more than who's working on the factory floor. You control the supply chain.
Okay, that is a really clear and powerful message. Now let's connect this directly to us, to human health. What does this mean for our own energy levels, our overall health? How much of our daily energy expenditure actually comes from this hidden microbial contribution? Is it a make or break amount?
Right. Putting it into the context of our own energy budget. The crucial question. The study provides some concrete figures based on that British reference diet. For that diet, they estimate the gut microbiota contributes about 4.4% of the host's total daily energy expenditure.
Okay, 4.4%. How much energy is that roughly?
For a typical adult, that's around 10 megajoules per day in total energy expenditure, so 4.4% of that. Now, 4.4% might not immediately sound like a massive number compared to, say, the energy you get from a meal.
Yeah, it seems kind of small.
It might seem that way initially. But remember, this is a consistent background contribution. And these SCFAs aren't just generic calories, they have those specific vital roles, especially nourishing the gut lining and regulating systems. So it's not just the quantity, but the quality and function of that energy.
Right, it's targeted energy with extra benefits.
Exactly. And that 4.4% isn't fixed. It varies hugely based on diet, just like the total harvest varies.
So what's the range?
It could be as low as about 1.7% in some individuals on very low fiber Western diets, those at the lower end of the U.S. NAE cohort, for instance.
Wow, less than 2%.
But it can surge to over 12% for those consuming non-Western traditional high-fiber diets.
Okay, 1.7% versus over 12%. That's a massive difference, like a 7-fold difference in how much energy your microbes are contributing just based on diet.
It's a huge range, and it really underscores how much our food choices influence this fundamental metabolic partnership. The study also makes a really important comparison, one that's critical for interpreting a lot of microbiome research we hear about. They compared humans to mice.
Ah, yes, the lab mouse model, used all the time.
All the time. And they made a key observation. Germ-free mice, mice bred with no gut microbiota at all, extracted significantly less energy from their food compared to normal mice.
Okay, so microbes definitely help mice get energy.
Yes. And that energy deficit in the germ-free mice was directly explained by the absence of fermentation products. No microbes, no SCFAs, less energy captured. Makes sense.
Yeah.
However, and this is the crucial point for extrapolation.
There's a but.
There's a big but.
Yeah.
The percentage contribution of the microbiota to daily energy needs in typical lab mice, fed standard, often autoclaved, chow, is much, much higher than in humans. It's often estimated to be over 21% of their daily energy demand.
21% in mice versus maybe 4 or 5% typically in humans, potentially up to 12%. That's a big difference.
It's a very significant systemic difference.
So what's the implication of that difference? When we read a mouse study about the microbiome and energy or weight gain, what should we keep in mind?
Well, if we connect this to the bigger picture, it means we need to be really quite cautious about directly extrapolating systemic energy effects observed in mouse studies straight to humans.
Because we rely on that microbial energy boost much less overall.
Exactly. Our overall energy metabolism is far less dependent on microbial fermentation than a mouse's is, based on these findings. Mouse studies are incredibly valuable, don't get me wrong, for understanding basic mechanisms, how microbes interact, effects within the gut environment.
Local effects.
Yes, local effects, cell interactions, immune responses in the gut might be more comparable, especially when you adjust for surface area, perhaps. But when it comes to the total body energy balance, there seems to be a fundamental difference. So it's just a crucial piece of context to remember when interpreting research findings.
That's a really important nuance. Context is key. Okay, this has been incredibly detailed and insightful. Let's try to do a final quick recap of the main takeaways from this deep dive into our gut's hidden power source.
Sounds good. Let's summarize the key points for everyone. First, your gut microbes are fermentation powerhouses. They take complex carbs you can't digest, mainly fiber, and turn them into short-chain fatty acids, or SCFAs.
Right. Acetate, propionate, butyrate, mostly.
Exactly. Second, these SCFAs are more than just energy. They're vital fuel, especially for your colon lining, and they act as crucial signaling molecules influencing immunity, satiety, gut-brain communication, the whole shebang.
Multitasking molecules.
Definitely. Third, and this is perhaps the biggest takeaway for practical application, diet is the primary driver of the total amount of these beneficial SCFAs you harvest. The amount of fiber and complex carbs you eat is the main control knob.
Not so much the specific types of bacteria.
The specific types influence the ratio of SCFAs, more butyrate here, more propionate there. But according to the study's framework, they don't significantly change the overall quantity nearly as much as diet does.
Okay. Diet determines volume, microbes determine flavor profile.
Good analogy again. Fourth, linked to this, modern Western diets generally lead to a lower SCFA harvest compared to traditional fiber-rich diets. This highlights a potential downside of our modern eating patterns for this specific aspect of health.
A consequence of low fiber intake.
It seems likely. And finally, remember that crucial difference between humans and mice. We rely much less on our microbiota for our total daily energy needs compared to lab mice. So be cautious extrapolating systemic energy findings directly.
Got it. Crucial context for research.
And of course, like any study, this one has limitations. The researchers are upfront about it. They used a somewhat static model of the microbiome, meaning it doesn't fully capture dynamic shifts over time. They didn't explicitly model all the complex cross-feeding interactions between microbes, especially for things like lactate. And they focused on the major fermentation products, not necessarily every single minor, but potentially impactful molecule like hydrogen sulfide or TMA.
Important to acknowledge the boundaries of the research.
Absolutely. Transparency is key. But even with those limitations, the rigor of the quantitative approach and the consistency between their independent methods make the core findings about the importance of diet and the scale of SCFA production really robust. This kind of quantitative systems-level analysis is exactly what we need to really understand these complex biological systems.
Yeah, it really moves the needle beyond just saying fiber is good. It starts to show us how good, why it's good, and how much difference our choices make at that microbial level. This deep dive genuinely highlights just how intimately connected our diet is, not just vaguely to health, but specifically to these microbial co-pilots and directly to our fundamental energy levels and systemic well-being. It's a powerful cycle we often ignore.
Absolutely. And moving beyond just identifying molecules like we discussed to actually putting reliable numbers on these metabolic fluxes, these flows, that's crucial. It helps us build a much clearer, more mechanistic picture of the links between our microbiome, these specific fermentation products, and maybe even the development or progression of various chronic diseases. This kind of knowledge really empowers us, right? It points towards clearer pathways for effective dietary interventions.
It really does. So here's a final thought for everyone listening. Next time you're planning a meal, or even just grabbing a snack, maybe pause and consider this. Are you consciously feeding your gut microbes the complex carbohydrates, the fiber they need to effectively fuel you back?
Are you providing the raw materials for your internal factory?
Exactly. What small change, maybe just one thing, could you introduce or increase to boost that vital fermentation product harvest? What stands out to you personally about how much your own everyday diet might be shaping your gut's hidden energy production right now?
It's a profound thought, isn't it? Your dietary choices aren't just about you. They resonate down into this vast microbial community, impacting a hidden energy source that has ripple effects throughout your body. Definitely something worth chewing on.